Cool. I want to move it. There. It might block some things, but we'll get over it, right? Okay. So my name is Cindee, and I'm from the University of Michigan. I teach in the departments of ecology and evolutionary biology, but also another program called CST, or Comprehensive Studies, for students who are underprepared for college in general. The students in that program are generally from either inner cities in Michigan, so I have a lot of students from Detroit, or from the Upper Peninsula, so they're very rural, so I have a sort of bimodal population of students. So I'm going to tell you a little bit about, I don't know, a course redesign that we undertook a few years ago that really, it was when we changed our book choice, and then we decided to really go very, very digital with a lot of delivery of content. We sort of are in a partial flipped, highly active learning classroom design, and I think it's worked really well so far, so I'm really proud of it. Thankfully, I think Sandra did a lot of the quizzing stuff before, so I'm going to skip a lot of that stuff. So about the course, we have about 600 students per semester. This is a course for students who are majoring in the biological sciences. So these are really, really motivated students, and they will take all of the practice they can get. They are primarily freshmen and sophomores, so even though they come in really interested and really motivated, they don't necessarily have great skills. So really structuring their out-of-class and their in-class time in a very constrained way was something that was really important for us. So this is the first in a two-semester course that's our introductory biosequence. So we often have first semester freshmen who just arrive and have zero idea what they're doing. And they are lovely, and I care for them deeply. The second course in the major sequence does the small biology. So we do the big biology. So we do transmission genetics, evolution, ecology, and then lots of work on science process, and lots of work on science process skills, and also sort of human impacts on evolution and the environment, and then a special focus on infectious disease, because that's just sort of, I don't know, in a disciplinary way, the interest of a lot of our faculty, so that works for us pretty well. So the way that we kind of conceptualize the redesign is having three pillars. So our first pillar is transparency. So before, when we used to teach a class, what's wrong? Okay, so when we first conceptualized the course redesign that we did in fall 2014, we thought it was really important to slim down a lot on our content and think a lot more about being transparent to students about our objectives, and then supporting those objectives with good practice. So we have three kind of pillars, right, that support our course and student learning on our course. So first is transparency. When I first started teaching, I thought it was part of the magic of teaching that you would make it a mystery about what you wanted them to learn. As it turned out, that doesn't work at all, right? So I thought, won't it be too easy if we just tell them what we want them to learn? No, not at all. It's still really hard for them. Hooray! So we tell them exactly what we want them to know by really detailed learning objectives. So every lecture has a set of, I don't know, 10 to 15 really detailed learning objectives that are easy to assess. And that makes it really obvious to our students what we want them to learn. It's incredibly comprehensive. And then they were like, oh, now I know what to learn. This is good, right? So we have reading guides that are prose written by the instructors that essentially tell the students, in the context of the book readings, what it is that we're interested in them getting out of the reading. So when students read a chapter in a textbook or even a section or a subsection, they're often like, what? How does this relate? So we've just written these like 2-page reading guides that are prose that say to students, oh, in this section, what we're hoping for you to get out of it is blah, blah, blah. Along with that, we have really targeted reading. So How Life Works is a slimmed down textbook already. And then we slim it down even more by assigning very small chunks to make it easily readable and sort of a more manageable chunk of work for students so that they're not feeling totally overwhelmed. And then lastly, this is like Mickey Mouse, and I was surprised. But we have vocabulary lists, and I was like, that's really lame. They really like it. For the students that are really struggling, starting with the basics, if you give them a place to start, it's really useful. In terms of concepts, so I've been teaching intro for 12 years. I teach half to a third of what I used to teach in terms of content, and way more deeply and with more support. And then our third pillar is practice. So for the practice, before lecture every day, they have a quiz that's due. And on that quiz, there are 5 reading questions and 5 what we call concept questions. The reading questions are really low level. Did you do the reading? Did you understand anything that you read before you came? And then the concept questions are exam style questions. So the other instructors and I have written a bank of around 1,000 questions that we put into LaunchPad. And so students are getting repeatedly quizzed on that. And so the concept questions are things that are like, from the previous lectures in this unit, do you understand? Are you able to synthesize? Are you bringing things together? Graph reading, stuff like that. We also have developed some visual synthesis activities for discussion sections. So students attend an hour-and-a-half-long discussion section with a TA every week. And so these visual synthesis figures in the book, that I'll show you one in a little bit, are really kind of synthetic and allow for us to use that as like a jumping off point for an active learning activity and discussion section. And then we have optional homework sets that students say by the second exam, they're not really optional, they're just not worth any points. But there's a lot of opportunities to practice, which is really useful.
Oh, click, I think she just click on there.
Yeah.
There we go. Okay. So like I said, when we redesigned the course, we have these pre-lecture reading guides, detailed learning objectives, quizzes due before each lecture, right? And then these discussion activities that support some of these higher order objectives. So getting them really into critical thinking and more application, more science process, and then harnessing some of the digital content that's available in the book that we didn't have before. So this is what a reading guide looks like. If you're curious, there's like a little checklist here at the top that tells them which parts to read, the vocabulary list, and the learning objectives for one lecture. And then the prose is this. So 3A, right, as described by the cell theory, right? All these cells come from pre-existing cells. Read this section to learn about how eukaryotic cells divide, right? And so this gives them, as they're reading the readings, really like a step-through kind of narrated guide. And these took an enormous amount of time to put together, and I thought the students were going to hate them, and they love them because the mystery is gone. So before we had these reading guides and stuff, students were like, too much reading, and I don't know what is important, and I don't know how to do it very effectively, and now we don't have that comment anymore. We also customized the book in LaunchPad into these lecture units. So for each lecture, there's a unit, and I'll show you what that means. And basically, we reordered the sections in the chapters so that everything the students need to read is read or do, which includes the reading guides, includes videos, includes supplemental materials, as well as the textbook readings are all organized into this unit. And then the students page through that when they're reading before they take their quiz, and then they know that they've done it. So before we had these, students were like, you're jumping all around the textbook, it's so confusing. Gone. We still jump all around the textbook. They don't find it confusing anymore because it's all in the same place, which makes it really easy. So what a lecture unit looks like, so this is my LaunchPad page from winter, and so this is a lecture unit, right, for each lecture, and then you can see the quizzes that are interspersed. So they essentially do the lecture unit, and then they go right into their quiz. So when I expand one of those lecture units, here's my coats to meiosis, right? Here's the reading guide, the introduction, cell division, et cetera, some animations, whatever. And when they're reading, it's not like it has to go, like if we were going from section 11.1 and this was chapter 28, section 2, it doesn't have to go through all the other chapters. It just goes automatically there. They love it. It's like crack or something. One semester when we were first getting it together, we were sort of behind, and they were like, come on, we need this. Kind of crazy. So that's our sort of LaunchPad course setup. Like I said, we do the quizzes, and similar to Sandra, we really like the question picker. We really like that you can copy quizzes from one semester to another. We really like the analytics on the quizzes. And so we do these quizzes all the time. And the student feedback on these is we hate them and know how good they are for us. We are eating our broccoli, and we know it's good for us. Thanks, we think. So the frequent quizzing is really important for us because it's a lot of formative assessment. It's worth a very small amount of points. And the best part about it is that students find out really early on what they don't know, so it doesn't take them until an exam. It takes them, you know, the first week of class, and they're like, oh, as it turns out, I'm kind of screwed already, huh? And so they start to come to office hours way earlier because they don't feel weird about coming to office hours to just say, I don't get it. They come and they say, I don't get this. Can you help me with this? So this speciation thing that we talked about, get that. Because this is like a really kind of basic question about what speciation is, and I chose this one, and as it turns out, it's all of them. Can you help me? And so having the quizzes frequently, right, and having both levels of questions means that students are really able to accurately reflect early on what they do and they don't understand. Then what we do before each unit exam is we take the hardest questions from all the quizzes, we put them into a giant bank, and we have students do what we call practice quizzes. These are no stakes, 0 points. They can do them as many times as they like. Call them practice quizzes. And they're sort of like an extra practice exam. We tell them to do them without notes, and then they have more questions to ask at reviews and things like that.
And you don't repeat those questions later?
Nope.
They're just, okay.
No. So we spent one very hellish semester writing 1,000 questions, and now we don't need to repeat.
So the real hard questions you took out? Or the questions you took out you said were real hard? Were you being?
So the questions that are on the practice quizzes were the ones that they had the hardest time with when they actually did their quizzes for a grade. When they do the practice quizzes, we just put all of the hardest ones on there, so it's much, much, much more like an exam. So it's a better studying tool because none of the low-level reading questions are there. You know what I mean? So we decided to track their behavior on the practice quizzes, which was a little hard because they're not actually, because they're not scored. It's really hard to track. So Elaine from Macmillan helped us, and so I did like a little first cut on the data analysis, and I was excited to see this. So if you did 0 to 1 practice quizzes, 2 to 50, or 51 plus over the course of the semester, so over 50 is an average of 12 to 13 practice quizzes per unit exam. If you did more than 51, your exam average is significantly higher than if you did 0 to 1 or 2 to 50. However, most students were not doing this, right? So this is our group of students who are going to do everything 63 times, right? That doesn't mean it's not helping this group, it's just not helping them in a significant way, but, I mean, this is a really big jump. So it's certainly not cause and effect, but I do think that the practice is something that's really effective for us. So we're kind of curious about how to look into this more deeply and see what our students are really doing.
But what was the N on that for the 51 plus? Because of those students that would have, because those maybe some of those could have been students who would have done, with no resources, would have been just fine.
Right, so this is 100 students did this.
Okay.
This is 300-ish students that did this, and this is 150 that did this.
Oh, I see the okay, hold on.
Yeah,
I missed that.
so this is about 600 students. Okay.
Okay.
Just wanted 2 y-axes, you know? So we have this whole course set up, right? These transparent learning objectives, this frequent assessment, and that was really good. And then we started to really harness the technology by using the visual synthesis activities, or visual synthesis figures from the textbook to build these discussion activities. So we've done this with 5 or 6 of the visual synthesis figures in the book. We're looking to do it more in our 2nd edition. But what a visual synthesis figure is it's something that the authors have built within How Life Works to take many disparate topics from different chapters in the book, right, and bring that together in a synthetic way. So if you want students to do the kind of higher order thinking that's required when you're approaching a novel problem, then you need to actually provide them an example of how to navigate through that. So I chose this one because it's my favorite and because I think it resonates a lot with the students because it's about breast cancer. So the BRCA genes are the genes that are known to be sort of responsible for inherited breast cancer in humans. And so we start out in the visual synthesis figure at sort of the big picture genetics level, but we also, you can get into the tumor, the mutant cell, healthy cells, right, all the way down to the chromosomal level. So the visual synthesis figures scale really well, which is important in biology from, right, subcellular level all the way up to a whole organism, which is important for us. So the thing that we do with this figure is we talk a little bit about, in discussion section, what a pedigree is, how it works. This helps us anchor for students that genes are not sort of abstract entities, but rather they're actually located on chromosomes, and so we do some chromosomal mapping. And then it also helps them connect something called meiosis, which is how we make gametes, to actually what happens when people have sex and they have babies, and those babies have genetic disorders that their parents gave them. So we kind of zoom in and out on the pedigree. So this is Ben and Sarah. So we take a close look at Ben and Sarah, and we make some predictions about what kind of alleles Ben and Sarah's babies would have. We draw a meiosis out for Ben and Sarah, so we're connecting those cellular processes to the whole organism. It's really fun. We do things like Punnett squares that actually have chromosomes in them to like make them think about that. And this is the activity where students are like, oh, I get it, right? So they're pulling all the pieces together because they're working through a series of questions that really requires them to probe the map and figure out what's happening. And then we take it and we make it a little bit more complicated. And so rather than looking at one gene on one chromosome, we look at another gene on a second set of chromosomes, right? So we've made the complexity even greater. Then we take and we ask them what happens if we put another gene on the same set of chromosomes, so we work through linkage. So we're working all the way from some very, very basic principles, sort of like maybe things they would have learned in 7th grade or 9th grade, all the way to linkage and linkage mapping and frequency of crossover and all these really complicated and quantitative things, all in the same discussion activity.
Is it in this body class, or is that, okay.
Yeah. My dream is to make a digital version that operates within seamless integration portal, right? So where you don't have to do it in class, but you can, right? So similar to our things like our online or our practice homework, our optional practice homeworks, where we would have a version that operated totally digitally, so we could have all of those activities and then just choose the ones that we wanted to do in class. Now, when we do this in class, we actually printed maps. So Macmillan actually printed physical copies of the maps for us, but students, I would say 50% of the students use the paper copy, and then the rest of them are just using their laptop. So this is an example of how we take sort of a digital resource and move it into the classroom into a really active way and help students work through that really hard thinking. That is something I just don't think we could have done without those ideas and that support in advance. I think that is it. Oh, no. So did it work? Well, maybe. So we really felt like before we did our redesign, we were really low in terms of the level of the questions that we were asking students and things like that. And so one Saturday morning, another instructor and I, we took and we bloomed 2 semesters worth of exams to see if there were differences. I would say that our blooming was not perfect because we basically just sort of back of the envelope were like, oh, what do you think that is? What do you think that is, you know? But this is what I wanted to show you. So in fall 2012, right, totally lecture-based, really content-heavy, no support, no learning objectives, maybe a few clicker questions here and there, right, because they're sort of level. Fall 2014, total redesign, learning guides, right, lecture units, all of that stuff, quizzes, everything. So our mean Bloom's level of questions that we were asking on the exams went from like a 1.5 to like a 2 or 2.5. So not awesome, because you still have to have some of those questions on there that are basic recall, but we're definitely significantly different in terms of the level of questions we were asking. Well, then the concern was you were making it too hard. There's no difference, right? They actually did slightly better, not significantly, but slightly better in the semester that's supposed to be harder. So we increased our expectations, right? We set up a system where we would allow for students to actually meet those expectations, and they did it. Now they're in Michigan, right? They can do anything. You can do anything that we ask of them, really. But this was really heartening for us to see. So one of my colleagues wrote a blog post about it. If you're curious about it, check that out. All right, so that's all I have. Did I go over my time? I didn't push the end recording button.